speaker is Eloise. The speaker is Eloise Frouin-Mouy. Eloise is an assistant scientist at the University of Miami and also an affiliate in the Passive Acoustic Ecology Program at NOAA Fisheries in Miami. She holds a PhD of biology from the National Institute of Scientific Research in Montreal, Canada. Before joining University of Miami and NOAA, Eloise worked for 10 years as a project scientist in the consulting company called JASCO Applied Sciences that specialized in underwater acoustics. Eloise is currently hosted at NOAA Fisheries in Woods Hole in the Passive Acoustic branch. So we have lots of time to chat with her if you have any more questions. Welcome, Eloise.
So hello everyone. So today I will talk about using acoustic in marine mammal research. So effective management and conservation of any species rely on adequate ecological knowledge of that species. Some of the key knowledge gaps in marine mammal research, especially pertaining to the conservation, can be addressed by understanding where the animals go, when they go, who the species go where, what they do when they are there, why they go, and how many are there. These questions are largely ecological because the distribution and the behavior of marine mammals are influenced by their environment, for example, by the ocean temperature, the ice cover, or the prey availability. However, the aquatic lifestyles of the marine mammals, along with their frequently hostile habitat, often makes acquiring this ecological knowledge problematic. So to overcome the challenges of studying these highly mobile species, a variety of tools have been employed. Those include the biotelemetry, the photo identification, the genetic, as well as the acoustics. With acoustics, we can use the passive acoustics. This approach is non-intrusive. It's behaviorally unbiased. We can use it for all weather or all visibility conditions. We can use it during day or night, and we can use it in remote areas. But to be effectively surveyed using passive acoustics, the study animals must produce detectable acoustic signals, and usually these must be identifiable to some useful category, for example, the genus, the species, or the behavior type. Passive acoustic monitoring of the marine mammals involves the reception of sound from a source, the animals, with a passive hydrophone, the recorder. We can also use the active acoustic. Active acoustic is a subfield of the marine bioacoustics involving the transmission of the sound and the analysis of returning echoes. And it's another tool that has been applied to marine mammal conservation, notably to look at the marine mammal presence and potentially their behavior. As with any research, the specific research question will drive sample design and the equipment that will be used. Here, I'm going to focus first on the passive acoustic monitoring, so the passive acoustics. We have three main challenges that are related to the site, so the spatial coverage, the time scale, and how many species you want to focus on, the diversity. I'm going to give you one example that is only with one species, the ribbon seal, but on very large spatial scale that include the Bering, the Chukchi, and the Beaufort Seas. And because we have so many recorders, even if we record during 1 year, the temporal scale was very large. Little background about the ribbon seal. It's a subarctic fossil species, and this one is endemic to the North Pacific. We know that they go on the seasonal pack ice from mid-March to mid-July, and after that, they are pelagic from summer through the late winter. On the map, you can see the light pink show the known extent of the distribution of the ribbon seal, and the purple actually show the pupping, breeding, and molting of the species. lucky for us, they are producing some distinct calls, the downsweep that I'm going to play. But as you can imagine, due to the difficulty of studying the ice seals in their natural environment, the distribution of the movement pattern of them over a large spatial-temporal scale are very poorly understood. So that's why we are using the passive acoustic monitoring. The objective of this study was to examine the migration pattern of the ribbon seal using some data, acoustic data from the Bering, Chukchi, and Beaufort seas. So it covers a large area. And it's a collaborative project, and so 4 different research groups provide data that has been collected in those areas from August 1st, 2012 to July 31st, 2013. And putting all the data together, we have 53 stations shown here on the map. We run on the station a downsweep detector that was created using a spectrogram correlation technique. And we did manual validation of all the detection just to figure out if the ribbon seals were actually present on the site. We found actually the ribbon seals were present on 30 stations, and those are shown here on the map. The area are color coded. So on the dark green, it's actually the Beaufort Sea shelf. The orange is the Western Beaufort Sea. The Chukchi Sea is in a pink and brownish colors. The Bering Strait is the light green, and the Bering Sea is shown as the purple. So those are the 30 stations with ribbon seal present. Here it's the same site shown from the south to the northern eastern. And you can already see that so the plot shows the presence at each day of the ribbon seal at each site, color coded by colors for the areas. The dates are at the bottom. On the y-axis, it's actually the ice concentration. It's the dark line that you can see here. And the gray means that we did not have any data at the time. So we can already see a migration pattern with some seals going from the Chukchi to the Western Beaufort and to the Beaufort Shelf, and then coming back to the Bering Sea, to the Bering Strait, and staying in the Bering Sea. Now I'm going to play an animation that you will see some recording sites. When it's a gray spot, it means that it's recording, and when you have a green spot, it means that we detect a ribbon seal on this day. So, and you can see also the ice coverage changing. So we have the ribbon seal mainly in the Chukchi, in the Beaufort during September and October. They are staying mostly during November in the Beaufort Sea. And then after that, coming down and staying in the Bering Sea from December to June. And you will see that we have more detection happening from February to June. It's because the matching period happens from April to May. And when the ice starts to leave, the seal left as well. And after June, we don't have any more detection from ribbon seals. Okay, so the passive acoustic, it offers some ecological insights regarding the presence of ribbon seal in the Bering, Chukchi, and Beaufort Sea. For example, we know that some ribbon seal were staying in Beaufort Sea and staying for a few days during in Bering Strait. So it means that it's probably ecologically important for them. But for Chukchi Sea, they were just passing through. So it's a migration corridor to and from Chukchi Plateau and Beaufort Sea for that. And the Bering Sea is actually the area where they do the matching. This study is actually important because it was the first description of a ribbon seal large-scale migration pattern using the acoustic detection, and we actually extended the known distribution because, as you remember, the pink was showing the non-extent, and on this site, on both sites in the Beaufort Sea, we actually had some ribbon seal detection. Another example is we were trying to apply the same idea for the hooded seal, unfortunately, so in the Gulf of St. Lawrence in Canada. Unfortunately, the underwater acoustic repertoire of this species is not very well known. So for this one, we had to do a little to actually record the underwater sound. So just some background. The hooded seal is a sexual dimorphic species. They are distributed through the North Atlantic and the adjacent Arctic marine areas. We have currently 2 management stocks here and here. Those two are not genetically different. And we know that they spend most of the year at sea. So the male actually can make sound in the air and underwater using the proboscis here and the septum. And I'm going to play a video that is that. So that is the bloop. The whoosh. It was a pink sound. So males are doing that to try to mate with a female. The female was not interested, not impressed by the male. So because, as I show for the ribbon seal, we can use passive acoustic monitoring using multiple recorders to measure the temporal and spatial distribution over large and remote areas. The idea is actually to improve our knowledge on the underwater acoustic repertoire. So far, only 2 publications was describing the hooded seal acoustic repertoire. And in blue, it's actually the sounds recorded underwater. So you can see that it was not a lot that has been described. And the 2 publications was from 1973 and 1995. So definitely we had to improve our knowledge of the underwater vocal repertoire before applying that for the passive acoustic monitoring. To do so, we went in the field to go near to the animals to record their sounds. And this fieldwork is why it's so difficult to access those animals and why only 2 publications exist. It's because the fieldwork is very short. Our ability to do the fieldwork, because it's related to the pre-FT of the reproductive season that happened just after the lactation period. The lactation period is only 4 days, is the shortest in mammals. So you can see that the seals go from this size to this size in only 4 days. So the milk is very fat. But because the lactation is so short, the reproductive period is short as well. And we need to access to the seal on the ice because we were landing with an helicopter. So we also need to have very good environmental conditions, so the ice condition and the weather condition. Lucky for us, in 2018, we were able to do it. So we had 4 days of data collection. That is the setup that I used. So we were using a microphone because I was also trying to record some sounds in the air. We have the ocean sound meters with the hydrophone recording the sound underwater. And I used a GoPro because I was trying to differentiate the sound that was recording underwater but produced by some animals at the surface versus some sounds produced really underwater. So using the classification and regression tree analysis and the rodent forest analysis, we were able to describe 17 underwater sounds. Some of them were already previously described, and those are not including the sound produced by the proboscis or the septum underwater. I'm going to play 2 of them because I like those. First one is the howl sound. And the second one is the hoo-ha. And yeah, they are produced by hooded seal. So looking at the data, so those were the sounds that, some of the sounds that are recorded during this fieldwork, and actually they match some call types that we have in previously collected data set that was collected along the Canada's east coast. So now that we have a better idea about the underwater acoustic repertoire of the hooded seals, we can now do the spatial temporal distribution of them along the east coast. And also, it's very important to share the sounds that you record on, because as you saw for the hooded seal, we didn't have any sounds from previous studies available to match the ones that we have potentially in our data. And so now the hooded seal sounds are on the DOSIT website. If anyone needs those, you can find them. So I talk about two passive acoustic technologies. The first one is the drop hydrophone that we use with the kind of drop hydrophone that we use with the hooded seal, and the bottom-mounted mooring that we use with a ribbon seal. But now I'm going to talk about another passive acoustic technologies, and this one is the acoustic tag. And I'm going to talk about a project that we do on the narwhal in the East Greenland. So it's a project that we recently started, so I don't have a lot of results to show you. But the idea is because we know that the narwhal use the echolocation to find the track and intercept the individual prey. Using the acoustic tag, it can act as an echo sounder. And from on this echogram that you can see here, we can see some echoes from some unsonified organisms that can be identified. And so it can help to better understand the foraging behavior of the endothocytes. So we were thinking that having the data from 4 female narwhals that has been tagged and scored by sound in Greenland in 2013-2016, maybe we could look at the foraging behavior using this approach. Unfortunately, those tags were placed too far back, and we don't have any echoes from the unsonified organism. But lucky for us, we have actually reverberation from the seafloor. So highlighted here in red, we can see more this one. And the echograms, I should say, that actually show the distance from the animals that has been tagged to the target. So for example, here you can estimate the distance to the seafloor from the animals. So the objectives for this project become to investigate the extent to which the narwhal females target the seafloor during the foraging, and to assess the diving behavior in 3D in relation to the seafloor. So that is for one of the females. Here it's the dive profile. So on the x-axis you have the time for each day, and on the y-axis you have the depth. It was a duty cycle recording, so we have low frequency acoustic recording in light gray, and we have the high frequency acoustic recording in black. And you can see on several occasions we have some high frequency acoustic recordings with actual seafloor reverberation, as highlighted by the section in blue. Looking at one of the dive profile from a female, so here on so this one is again an echogram. So you have a distance from the animals to, in this case, the seafloor, and I put on it actually the dive profile of his individuals. So looking at this information, we have the dive profile, but we can also calculate the seafloor depth on several occasions. The other information that we have from the tag is actually we can estimate the narwhal orientation during the diving. And for this individual during all the tagging period, we look at the rolling and the color code by the position of the body along the roller axis. So the red means that they were ventral up. The light blue means that they were dorsal up. So it makes sense when you look at the surface, they were dorsal up. If you put this information together, yeah, I know it's very ugly, but they are the only narwhal 3D models that I could find for free. So they are in the Microsoft PowerPoint actually. It's the only one that is ugly, I would say. But so on this one, same thing. So you have the echograms with the distance per seafloor, and here you have the rolling. And you can see that when the female is coming near to the seafloor, she's actually ventral up. And it happens on several occasions, here and here and here as well. So now we are currently looking at this behavior for the narwhal. Using another technology, this one is the float channel recorder or tracking app, tracking array. We use it for the big whales in the Gulf of Mexico in a project called the Listen-GOMEX. So Listen-GOMEX stand up for long-term investigation into the landscape, trends, ecosystem, and noise. And it's a 10-year project with a short and long-term side that you can see under the big whales in this one. And this, one of the objectives is actually looking at the trends, and to do the trends, we need to look at the density estimation. So the objective of my sub-study in this one is actually to advance the passive acoustic monitoring-based density estimation of big whales in the Gulf of Mexico. To apply the distance sampling approach for the density estimations, it requires some estimating, detecting, and acoustic signals of a Q given an animal's distance from an acoustic recorder. So here is the formula of the density estimation. One of the most important things, as I say, is to estimate the probability of detection. And the probability of detection of detecting a big whale click as a function of a distance from a single sensor is estimated by simulating clicks at a state of distance. And to do that, knowledge of a big whale source level and characteristic of the sound beam pattern, as well as some detailed population-specific information on subsurface diving, including the subsurface group size and group spread, and acoustic behavior are very critical information for an accurate detection probability estimation. Normally, those information, so acoustic behavior, diving behavior, are provided by tagging information. Unfortunately, we don't have any big whales that has been tagged in the Gulf of Mexico. So we had to look at another approach. And to do so, we are using the passive acoustic tracking and using 2 tracking apps. So it helps us to actually do the dive profile of the whale. By using the TDOA, the time difference of arrival, we can do detection, localization, and tracking in 3D of the individuals. So for this one, we were looking at one site, the Green Canyon, where we deployed 2 tracking apps, GCO1 and GCO2. Those were apart from approximately 800 meters. And we record 6 months of data from early September 2021 to late March 2022 at a sampling frequency of 100 kilohertz. So to do the 3D tracking, first we were using DataEdit to review and validate some detection and associate detection with big whale species. And after that, we were using Where is Weldo, that is a MATLAB package for some time difference of arrival localization. So in Where is Weldo, it's displayed like that. So the Where is Weldo. So we have for each of them, for each tracking app, so GCO1, we have the time versus the azimuth, and the time versus the elevation. And on this plot, it's the azimuth versus the elevation. So we have that for the both tracking apps. Normally what you do, it's you're labeling one of the arrays, so it's easy to differentiate the individuals based on the azimuth or the elevation. And after labeling one of the array with all the individuals, we can do a click-train correlation to associate the detection across the 2 arrays by aligning a series of detection with the same temporal spacing. and an approximate TDOA of each click train between the two arrays was estimated. So meaning that, for example, I labeling this one, I'm doing my association, and it's going to show me some, sorry, I'm going to use another example. I'm labeling the red one here, and doing the association is going to pop up some red points on this one, on the second array. When you are done with labeling the two arrays, you can actually do the 3D localization using the azimuth and the elevation of both arrays. And so in this case, we had 3 cuviers, and you can see the dive profiles of those on this animation. So some of the dive were actually rejected from the analysis because they represent some group of animals whose individual detection angles could not be unambiguously discriminated, and or because some track had too few detection to be reliably localized, and or because the track was too short, meaning less than 7 minutes. So on the 6 months of data, we were able to have 24 tracks for cuviers, 24 tracks for Gervais, and unfortunately only 2 tracks of Blainville because they are not very present at this site. So looking at the tracks, we could estimate the horizontal distance for each of the species, and we could also look at the azimuth distribution for each of them. And on this plot is the horizontal distance that you can estimate looking at the relative easting versus the relative northing. We could also look at the diving behavior. And so this plot shows the elapsed time, so meaning from the beginning of a detected dive. And this, the y-axis is the depth. So we can look at the descent, bottom time, and the ascent. For example, in the cuviers, we have some descent, bottom, and ascent for Gervais, but we don't have any ascent for Blainville because we have only 2 tracks for this one. What is interesting, actually, it's for the cuviers. You can see that several of the individuals were spending some time at or near to the seafloor, and the seafloor at Green Canyon was around 1,100 meters. So, and for Gervais, they were actually higher in the water column, meaning that the 2 species have probably a different diet. They are probably targeting different prey. And so it's something that we are going to explore a little more further. We could also estimate the source level and the directivity index for both the cuviers and the Gervais. So we did that using the off-axis angle. And the off-axis angle refers to the angle from between the connecting line to the acoustic receiver and the clicking wheel direction of motion. So that is your off-axis angle. When you have an off-axis angle of 0, it means that the animal is facing the acoustic track. 90 is passing, and 180 is facing away. Here, a radial broadband piston beam pattern, so that's shown as the solid line, was fed to the median of the source level distribution that is shown as the dashed line. And for the off-axis angles between 0 to 90 degrees, for the cuviers, so it was with a source level on axis of 225 dB and a directivity index of 26 dB. And for cuviers, it was 280 dB and a directivity index of 20 dB for the Gervais. Now that we have a better understanding about some acoustic parameters and diving parameters, we are doing a Monte Carlo simulation. So what we need to know for the acoustic parameters, it's the source level, the peak frequency, the minimum off-axis amplitude loss, the directivity index. For the diving parameters, we need to know the dive depth mean, dive altitude mean, dive depth max, the depth at start of clicking, and the descent angle. So we could obtain most of the information from this study. The information that we could not, we are still using the literature for that. Fortunately, is from species that are not in the Gulf of Mexico. And the Monte Carlo simulation is going to give you a probability of detection. So we were doing a comparison between the in situ, meaning the values that we obtain at GCO1 and GCO2 in gray and dark, versus the Monte Carlo simulation, and that we can see we have a good agreement for both Cuvier and Gervais, and obviously not for the Blainville because we have only 2 acoustic tracks. But at least for Cuvier and Gervais, now we can use the Monte Carlo simulation to do the density. We are going to use this approach to do the density estimation. Another technology is the acoustic drone that I developed and test for gray whale in Baja California in Mexico. And so the idea is because we know that the marine mammal use both the vocal and the non-vocal sounds for acoustic communication. We were trying to, one of the things is to better determine the sound function. We need to have the calling individual's behavioral context that is measured at the same time that the sound production. So we need to know the immediate behavior state. We also need to know the identity of a sound producer as well as the group size. As you can imagine, it's very challenging, both logistically, but also because associating some vocalization that normally happen underwater with some specific behavior that could happen underwater or at the subsurface is not easy. So we were wondering if we can use some drones for the marine mammal acoustic research, and we did a case study with a gray whale with the Eastern North Pacific population. So just to let you know, this population is coming to calving and breed in some lagoons in Baja California, and the one that we went to the fieldwork was the Laguna San Ignacio. There, so we were using 2 drones. One drone was the DJI Phantom 4 Advanced Plus. This one was used at the video drone, so it was recording some activity of the whale. And the other drone was the Swell Pro Stratron 3 Plus, and I attached a sound trap, the 300 STD, using a rope. And when it was landing on the water, the recorder was 2 meters below the surface. Because I was on a budget and my drone was not floating very well because of the payload, I had to put some foam noodles, and also at the time, this sound trap doesn't have any cage, so I put some wire loops from the kitchen whisk. Worked very well. Thank you. Here it's the display. So we have a video drone taking videos, and we have an acoustic drone that we can land actually very close to the whale to record the sounds. Here it's the acoustic drone actually flying above a whale, and so you can see the sound trap here in the air. And due to some technical issue with the drone, we had to limit our distance from the coast to 300 meters. That was actually totally fine, because most of the time it's where the mother and calf are in this area. So on 7 occasions, we were able to fly both drones at the same time. And so we have simultaneous record of videos and acoustic. So those are the 7 flights that we were able to do. Here you can see the location of the San Ignacio Lagoon and the Cuenca Piedra location where we were doing the fieldwork. And I'm going to play a video, and I'm going to explain it first. So you will hear first a croak sound, and it's a mother making this sound, and after that she's going to do a bubble blast, and the calf is on the top of a bubble blast, is rolling in it. So she just say, "Hey kitty, here, it's time to play." That is the croak sound. And here you can see the calf rolling in the bubble blast sound. Because I want to show you where is the drone. So here we know where is the female, and so here we know where is the female, and here is actually the drone. So because we know the distance using the same technical approach that the photogrammetric calculation, we can estimate the distance from the source to the receiver. And because we know the sound that she produced, we were able to estimate the source level of the croak and as well as some bubble blast. Because we were so close to the whales, we were able to record some very quiet whale sounds that were not previously described in the literature. So maybe those are new sounds or new call type. And obviously, this approach is only suitable for some shallow and clean waters because you want to be able to observe the behavior of the whale. And I will conclude with another technology that is the deep sea cable observatory. And for this one, we use both the passive and the active acoustic technology. And we did it at Barclay Canyon in Canada, and we were studying the northern elephant seal. So the northern elephant seal, it's the only mammal that is doing migration twice a year. So by traveling into the North Pacific in a pattern that we call the double migration. And as you can see on this map, the information, the biological information is highly biased towards the female, and it's because it's due to the higher survival rates, but also due to the easier handling to capture them and put some instrument on them. So we have way less information for the males, and notably for the sub-adult males. So, for example, we don't really know where they are foraging, what they are foraging on, and the length of their foraging trips. From May 2022 to July 2023, a fish acoustic and attraction experiment, or FAAE, was actually taking place at Barclay Canyon, Barclay Node here, and also shown as the green star here. And at this site, a deep sea cable observatory with HD video camera, Ditson sonar, that is an acoustic camera, and the hydrophone was used. So here is the setup. You have the sonar here, you have the video on the top, and we have two LED that was turning on just before the video start recording. Here is the field view of the HD video camera. You have a bait dispenser here, and you have the hydrophone. And similarly, this is the field view of the sonar with a bait dispenser here and the hydrophone. So this site, this experiment was focused on the fish activity at the site. But lucky for us, we received some unexpected visit from sub-adult male northern elephant seals. This one is very slow. And so just inspecting the area. And because we had this opportunity, we decided to use it to better understand the deep sea behavior of the sub-adult male northern elephant seals. So we can see them on both the HD video and on the sonar. So as I say, we have a hydrophone. This one was recording continuously. We had the Ditson sonar that was recording for 15 minutes every hour. And after the Ditson sonar starts, 5 minutes later, the light turned on and the HD video stopped recording for 5 minutes and then stopped. And the Ditson sonar was still recording for another 5 minutes. So we have the before, when it's only the sonar, the during, that it's the sonar and the HD camera, and the after, that is only the sonar. And each of those conditions are 5 minutes, approximately 5 minutes long. Looking at the Ditson videos with northern elephant seal presence, we have 210 of them. So looking for each condition, the before, during, and after, we saw that in the before, we have actually several individuals that were resting. And when I say resting, it means that the individual was at the sea bottom, motionless, taking a power nap, I guess. But you can see that during, for the next condition, the during, they were not here anymore. And it's because when the light turn on, it's waking them up, and they just left the area very gracefully, as you can see. And sometimes we could hear them touching the hydrophone when they were leaving. But on one occasion, actually one of the individuals slipped through, mostly through, and because of the body orientation, he was facing away from the light, and so that's why you can see only on the HD video and also on the sonar the flippers. This one was sleeping for at least 8 minutes and 40 seconds, and when I say at least, because it was already there when the sonar turned on, so maybe it was there for a little longer. When they were not resting, they were actually foraging, and they were mainly focusing on sable fish. That was the most common prey, or potential prey, I should say, at this site. Interestingly, on several occasions that we saw when they were foraging, they are producing some head bobbing. And on many occasions, when they are producing their head bobbing, we actually record some low-frequency sound that you can see here on the spectrogram. And it seems that the head bobbing and the low-frequency sound associated with them induce a startle response in the fish. Normally, the startle response is pretty immediate. In this video, it's a little delayed. But we think that the northern elephant seal, at least the subadult, use this technique, this approach to improve the foraging success because it's easier to follow the fish when it's moving. Looking at, I named the seals. Looking at the daily occurrence, so here on the bottom, it's the date. And on the y-axis, it's the hour in local time. So you can see when the different identified individuals. So we have 6 that I call frequent visitors that came more than once. And so we have Brian in blue, we have Dennis in red, Carl in orange, Mike in green. Blondie was saw only twice in yellow, and Bruce was on several occasions at this period. When it's dark gray, it means that we have northern elephant seals, but they were saw only on the Ditson, not on the HD. And when it's in black, they were saw on the HD, but we could not identify them as an individual. Interestingly, so we saw that Brian, Dennis, Carl, and Mike came on period over 10 days. With Mike, he's been having a record of 30 days. But after that, the 2 other individuals that came more frequently came only on 1 to 2 day period. And so it also corresponds when the Ditson sonar failed. And looking at the acoustic, we found that the Ditson was making sound, producing some noise when it was scanning, but also when it was on standby, as you can see here on the spectrogram. And when we put the information of the noise for when the Ditson is on or when it's off, and it's actually in the hearing range of the northern elephant seal audiogram. So we found that at least 4 of the northern elephant seals were able to locate precisely the site for a long period. So as I say, over 10 days, possibly due to the noise generated by the sonar, suggesting that they learn to use the anthropogenic noise as an indicator of food location, also known as the dinner bell effect. So to conclude, I present you 6 case studies with a ribbon seal. We use the passive acoustic monitoring to better understand the migration pattern. For the northern elephant seal, we use active acoustic actually to better understand the behavior at depth of the subadult males. With the gray whale, we develop the acoustic drone to better understand the behavioral context. For the narwhals, we are using echograms to look at the diving behavior at the seafloor for these species. For the Odyssey, the idea was to record the underwater acoustic repertoire to later use it for the passive acoustic monitoring. And for the big well, we are looking at the 3D tracking so we can improve the density estimation in the Gulf of Mexico for those species. If you are interested in passive acoustic technologies, we have a book chapter that is going to be released normally at the Marine Mammal Conference in the book titled The Marine Mammal Research in the 21st Century, Emerging Technologies and Applications for the Field and Laboratory. And I would like to acknowledge all my collaborators on those different projects because I was not the only one doing this work. And thank you for your attention.
Thank you very much for the very interesting talk. I was very fascinated by the 3D individual tracking of the narwhal that they have belly up, and that I had no idea what they were doing. So it was very interesting. We have some time for questions. Oh, we have a Zoom people, so yeah.
Hey, Eloise, thank you. It was great to see all the stuff you've been doing. I was just wondering how you knew that these were juvenile male elephant seals in that project specifically.
Yeah, so you can determine that they were subadult male based on the shelf chest shield, and the development of a proboscis. So it's how you can estimate the age. Actually, we could estimate the age of each individual. It was ranging from 4 to 7 years old.
Any more questions?
In the case of the acoustic drone, does the sound of the drone itself get transmitted down into the water at all?
Good question. So the good thing with the drones, this drone, it can land on the water and you can turn off the rotors, so you don't have any sound from the drone when it's actually land. But it's actually have a water surface, so you don't have any noise from that.
Any questions from the audience?
And in the case of the beak whales, how could you tell which species they were? Maybe you said that and I didn't catch it.
To know which species it was making so each beak whale have a very distinguished acoustic signature. So that's why we can identify them by species based on the ICI and on the acoustic signature.
For the beaked whales, have you looked at the prey field at the same time? It looks like they're consistently going to the similar depths.
Yeah, so not yet, but it's definitely on something that we want to explore a little more because we want to try to better understand why the Cuvier is going at the sea bottom when, for example, the Gervais is not.
Thank you for a really nice talk. I'm curious, with all the observations you have and knowledge you learn from those kind of observations, are there applications? Are there examples that you use those knowledge for things like conservation purposes or some improving the ecological knowledge?
So, good question. So, for example, for the big whales, it's definitely to be for conservation and management because we try to better understand the trend of the populations of big whales in the Gulf of Mexico following the Deepwater Horizon oil spill. And so that's why we have acoustic data from 2010 from now to now, And so we want to go back to this data and use the density estimation to look at if actually the population is decreasing following the Deepwater Horizon or decreasing and now coming back, or just trying to better understand the trends. So yes, we have some application for that.
there any question on Zoom? Oh, one more round? No? All right. Well, thank you
Yeah.
so much, Eloise.
So one thought. I have one thought, if you can still hear me. This is Michael Moore.
Yes.
Just a comment, really. That in 1949, Bill Cheville and Barbara Lawrence made the first recording of belugas underwater. That's just 75 years ago. And here we are today with all of the acoustic work you're doing. It's a remarkable legacy, and times have changed, and they'd be thrilled to see what you've been doing.
Thanks for the comment.